Good morning, students. Today we are going to learn about strategies for enhancing in food production. So, in this lesson, what is the name of the lesson? Strategies for enhancing in food production and plant breeding. The name of the lesson is, see what are the traditional type of farming and what are advanced type of farming we are going to learn in this lesson. First of all, let us see what is what do we get through the traditional farming. Okay, through the traditional farming, when we see here the biomass of the plant, will be very less when we take as a food for the animals. Okay, when we go through the traditional farming. So, what is biomass? The biomass means the amount of organic matter, which is present in the plant body, that is called as a biomass. Usually plants and animals and human beings, we take the food from the plants. So, in that plant body some organic material is present, that organic matter we are calling as a biomass. So, through the traditional farming what happens? The biomass will be very less. So, in order to get more biomass or more yield, high yield or more productivity, we are going for the plant breeding. What is that? That is plant breeding. What is plant breeding? It is a purposeful manipulation of a plant to desired characters. So, we are taking one plant, in that plant, from that plant, or else if you want to get a new plant which has a desired characteristics, so for that purpose we have to go for the plant breeding. In the plant breeding there are some steps are there. So, whatever the steps, whatever the techniques we had, those we are calling as strategies. So, the steps, the techniques which we are following in order to enhance the productivity of the crop or the resistance of the crop, what steps we are taking, those all steps we are calling as a strategies. What purpose we are using these strategies, what purpose we are using these steps? To enhance, what we are enhancing? Food production. To enhance the food production, what steps we are taking, those we are calling as a strategies. Okay. In olden days, that farming is called as a traditional farming. Already we have seen the word traditional farming. In the traditional farming, when we see almost 9,000 to 11,000 years back, 9,000 to 11,000 years back only we had a traditional farming. So, wherever the culture and civilization has developed, there we can see the traditional farming. Wherever the water is available, there the farming has started. But thousands of years, what is that? Thousands of years back only the agriculture, the farming has started. Here, with the help of the traditional farming, what we don't get? We don't get a proper yield. Why? Because the population is more, India's population, all over the world population is more. So, whatever we cultivated, what yield we are getting, that yield not sufficient to meet the needs of this growing population. If you continue the traditional way of farming, then you may get the productivity, but that may not satisfies the food requirements of the growing population. And also resistance. What resistance? There are different type of the resistance are there, biotic resistance and abiotic resistance. What is biotic resistance? Biotic resistance means insect, pest, fungi, bacteria, virus. So, these microorganisms and insects both are there. So, through the biological organism, anything is affected to the plant body, the productivity of the crop will be reduced. Those comes under the biotic resistance. Whereas abiotic means light, that may be the light, temperature, water. Okay, these three comes under the abiotic resistance. If the amount of the water is less, then what happens? Ultimately, it affects on the productivity of the crop. So, that is if the water lagging is there, then definitely the crop production will be reduced. So, nowadays whatever we are producing, the seeds or the plants or the hybrid varieties, these all are possess a resistance based crops. So, now compared to the traditional way, hybrid way or whatever we are using artificial hybridization, modern technology aspects, steps whatever we are using, those are fulfilling the needs of the growing population. Then how do we get that? Let us see. So, now if you see advanced technology, through the advanced technology, for example here molecular biology. What is that? Through the molecular biology or genetics or plant breeding or tissue culture. What are these? These are different branches of the biology. With the help of these modern branches of the biology, those principles, we are getting hybrids. What we are getting? Hybrids. These all are artificially we are getting. How we are getting? Artificially we are getting. So, whenever we use such a artificial method or cross hybridization or hybrids, then what do we get? So, due to that, what do we get? Then we get a high yield. First of all, we get high yield. Next, high yield means the productivity, the crop production will be more. Next, resistance. Resistance for pest, insect, bacteria, fungus, viral, etc. Okay. So, through the artificial mode of hybridization, what we are getting? Different types, means different type of the resistance we are getting, or else stress resistance. Under the stress resistance, you can able to see two. One is salinity, that is a salt, another one is water. Nowadays, whatever we are producing the seeds or the plants, even some amount of the water is less, also they can able to survive. That is the new technology which we are adapting. Another one is the salinity, the amount of the salt which is present in the water. Due to that, salinity concentration increases in the water. When the salt concentration increases, it retards the growth of the plant, means the growth of the plant will be stopped whenever the salt concentration is increasing. So, first one high yield, second one resistance. In the resistance there are two types, one is biological, another one is abiotic, biotic and abiotic. Now, next one is fertilizer response. We use fertilizer, so that by taking that fertilizer also, it has to be withstand. Why? Because nowadays whatever the fertilizers we are using, these are the chemical fertilizers, organic fertilizers. So by absorbing that organic substances, the plant has to be withstand. That comes under the hybrid. Okay. So based on this stress resistance or else biological resistance, high yield, fertilization and more crop, these all are we are getting. No, that is the reason why we are calling as a hybrid. And soil fertility, based on the salt, soil fertility will be increases and decreases. So now we got a clarity between the traditional and the artificial or hybrid varieties. So the traditional biomass of the plant will be very less. Whatever we take food from the plant, the biomass will be very less. So that what happens, we cannot meet the need of our food diet. So we have to increase whatever the organic matter is present in that. Through this technique we can able to increase. Now to meet these needs of the growing population, we know that green revolution has taken place. Okay. So before going to learn about the green revolution, here there are some steps are involved to get the more productivity. Okay. And nowadays whatever we are getting hybrid varieties, these all are wild traditional one only. You may get doubt, what is the difference between the wild variety or the normal variety? See, wild variety means it is growing its own. We are not taking care of its, the plant or the seed, we are not taking care of it. It is growing on its own. Means whatever the nature is giving, the water or else the sunlight, from the soil nutrients and minerals, those all are absorbing with the plant or the seed and it is growing by its own. So those are all wild varieties. Okay. Then traditional, we can able to form them through the traditional one. Now artificial means we are by our own, we are supplying the nutrients, minerals, and we are incorporating some genes in order to overcome the problem of the biological stress as well as abiotic stress. So those all comes under the artificial hybridization. Now let us see what are the steps are involved in the strategies for the food enhancement. The steps involved in the strategies for the food enhancement. Okay. So let me write here first. The steps I am writing here. Collection of germplasm. First step is collection of germplasm. Second one is evaluate and selection of parental plant. Third one, cross hybridization among the selected plants, among the selected parental plants. Next one, selection and testing. Fifth one is testing and releasing into the market. These steps are very, very important. Based on these 5 steps, the question will be framed. Which of the following steps are correctly framed, or which of the following steps are incorrect? Jumbled, it will be jumbled and the question will be framed. So you have to remember line wise. Collection of the germplasm, evaluate and selection of the parental one, cross hybridization among the selected one, and selection and testing, and fifth one is testing and release. Now let us see one by one. Collection of Germplasm. What is germplasm here? The entire, that may be the plant or the seed which has the various alleles of genes of the total plant or the seed is called as a germplasm. Means it consisting of the characters, whatever the desired characters are there, round or else high protein content, sugar content, disease resistance, such type of the characters, those characters are under the control of the genes. These are present in the plant or the seed. So, we are collecting such a type of the desired characters which are present, such type of the seeds and plant we are collecting. So, that collection is called as a germplasm. Germplasm means just we are collecting the seeds which has the desired characters, that is called as a germplasm. And also germplasm contains, it consisting of, what is it consisting of? It consisting of cultivated, improved varieties. Next, improved varieties, no more cultivation. Third one, Deshi are local varieties. Next one, pure lines to the plant breeding. Next, wild varieties, from the wild varieties we are getting the hybrid one. So, what does a germplasm contains? What is a germplasm? It may be the plant or seed which contains the total desired characters. We are picking, we are collecting among the group. Next, what does it contain? Cultivated improved varieties, means whatever the improved varieties are present, that we are selecting. Next, improved varieties, no more cultivation, no more, that improved varieties we are preserving. We are not for the cultivating, of course for the next year cultivation purpose whenever we use, that is different, but a germplasm consisting of what are the characteristics? Cultivated improved variety, it has, the germplasm has cultivated improved variety and the germplasm has improved varieties. Deshi are local varieties. From our native place, from our crop land, agriculture land, what we have cultivated, from that crop we have selected some seeds and some plants which has the desired characters. So, that germplasm we are calling as here Deshi are the local varieties. Next, pure lines. Pure lines means homozygosity. For example, A and B are there. After crossing we will get AB. This AB again crossing with AB only. So, what we are doing, same gametes or same genes we are crossing. So, that homozygosity or else here same characters we will get. So, that is nothing but here pure lines. Next, wild varieties. Whatever the new desired varieties we obtain, those all got from the wild varieties. So, this is the first step which is present in the strategies. What is that? Collection of the germplasm. One more important thing is the germplasm will be stored at low temperature. This is very, very important. Where it will be stored? It will be stored at a low temperature. What is germplasm here? Seeds, collection of the seeds. Collection of the seeds is nothing but germplasm. The literary meaning, actually the plants which has the desired character, the seeds which has the desired character, all together entire which has more characters, desired characters, that is called as a germplasm. Okay, then whenever anybody asks where it will be stored, it will be stored at the low temperature. Okay, then here the germplasm, when we see the germplasm, it is a backbone for the building of the crop improvement or the plant breeding. Okay, germplasm, it is a backbone or else root for plant breeding, for cultivation of the crops or plant breeding. And also what we are doing, we are collecting, and what we are doing, we are preserving, collecting and preservation. Germplasm we are collecting every year from our crop agriculture land, among them which has the desired characters, those seeds we are picking and storing. For what purpose? For the future use purpose. So collecting and preservation for the future use, and germplasm is a backbone for the plant breeding. Okay, these two steps are very important. Where it will be stored? At low temperature. What is germplasm here? Seeds or the plant are called as a germplasm. Why we are calling seeds or the plant called as a germplasm? Because it has entire desired characteristic of that plant, so that we are calling as seeds are called as a germplasm. So now out of the 5 steps we have completed one. What is that? Collection of the germplasm. There is no doubt regarding the germplasm. Germplasm is nothing but entire desired characters consist by the plant or the seed. Where it will be stored? It will be stored at the low temperature. What is the germplasm consisting of? Cultivated improved varieties, varieties, no more cultivation, and Deshi are local. From where we are collecting those varieties? From our local areas only. Then pure lines. Pure lines means after hybridization, what we got a hybrid, again and again we are crossing with the same character, that is called as a pure line. Homozygosity will get. Then wild varieties. What is that? Wild varieties. We have not. Nowadays we are cropping and we are forming, but once upon a time they have grown up their own, so that whatever the hybrids we got, those are all wild varieties only. Now let us see the second step, that is, this is completed. Evaluate and selection of parental one. So second one, evaluate and selection. Selection of parental plant. Okay. So now what we got? A desired germplasm we got. So now what we are doing? Total germplasm. Germplasm is nothing but what we have learned, seed or the plant. Total germplasm we are collecting now. Total germplasm we are collecting. After collecting total germplasm which has desired characters. Desired characters germplasm only we are collecting, no? Then after, from that we are dividing into 2. Why? Because some here bisexual flowers plants will be present. So whenever the bisexual flowers are present or plants are present, what we are doing? We have to label them male plant as well as female plant. When in the case of the bisexual plants only, we have to label like a male plant and female plant. Whereas in unisexual flowers or plants are there, then you need not to label. Why? Unisexual, either male or female only present, so that you need not to label. By seeing the flower, you can able to understand whether it is a male or female, so that you need not to label for the unisexual. Bisexual means we have to select this plant is a male plant and this plant is a female plant. Like that we have to select. So what we are doing in the first step, just we are collecting the germplasm. What type of the germplasm? Desired characteristics having. So what is germplasm? Either seed or plant we are treating as a germplasm. Next, second step, evaluate and selection of the parental one. We have collected the many seeds, those are the or else plants we have collected. Among those plants, some of the unisexual are present, some of the bisexual are present. Unisexual you need not to label, but bisexual we have to separate. Okay, we have to evaluate. Evaluate means this one we are taking as a male parental one and female parental one. So, we have labeled like that, male plant and female plant. Okay. Now, second step also completed. So, this is what evaluate and select. From the germplasm we have evaluated. Selection, what we have selected, this is a male plant, this is a female plant, like that we have selected. So, now what we have completed? Second step also completed. Next, third step, let us see. What is third step? Cross hybridization among the selected parents. What is that? Cross hybridization among the selected parents. We have selected in the previous step male and female. Am I right? So, now let us see. Now, just I am writing A is a male or B is the female. Okay. Then A consisting of some character and B also consisting of some desired characters. Okay. Whatever the desired character possessed by the A, that desired character may not present in the B. What desired character possessed by the B, that may not present in the A. Okay. So, now these two parental desired characters we want to see in one plant. So, that is called as a hybrid one, superior one. Okay. Now, this A plant consisting of more protein, but whereas resistance, what resistance? Disease resistance. The resistance, which resistance? Disease resistance is less, but highest protein content is present, disease resistance is less. But whereas B plant, when you see, the protein content is less, then disease resistance is high. Here disease resistance less, here disease resistance high, here protein content is high, here protein content is less. So, what are the desired characters? The protein content is a desired character. Here what are the desired characters? One is the disease resistant is the desired character. These two we have to see in one. So, when you do the cross hybridization, what do you get? A and B. This is only we are calling as a hybrid. What is hybrid? In the previous lesson we learned hybrid means it is a mixture of the two parental one. Okay. Now, I am calling as this one superior recombinant. Superior recombinant. Why superior recombinants? Either of the parental or both parental characteristics it is possessed, so that it is called as a superior recombinant. But this hybridization, cross hybridization, when we take, it is a most time taking process, most time taking process or tedious. Why it is a most time taking process or the tedious? Why? Because in the group of plants, when you take, here what happens? The desired gene, the desired chromosome, may be present or absent. If it is present, it undergoes into the hybridization. if it doesn't possess, what happens? You won't get the hybrid variety. Okay, naturally when you take, what happens? It may possess the chromosome, desired chromosome or desired gene, or may not possess desired chromosome. That is the reason why it is most time taken and tedious. After the hybridization, if you not get the superior recombinant, what you have to understand? Because there is no desired characteristic in that plant or in that seed, that is the reason why superior recombinant we did not get. So that it is the most time taking and tedious job. And another one is, out of 100 or 1000 of the seeds of the plant, only one will be performed this one. Out of 100 and 1000 cross pollination, you may get one superior recombinant. 100% we cannot say. Definitely through this pollination, cross fertilization or the pollination, we may get superior recombinant like that. When it occurs naturally, I am talking about naturally. When it occurs naturally, we cannot say 100% superior recombinant production. Okay. Then here chromosome may be present or absent. Out of 100 or 1000, only one will be possess this superior recombinant. Okay. So due to this above reasons, cross hybridization is a time taken and tedious. One more also I am explaining. Whenever you take artificial hybridization, for example, let us assume through the artificial hybridization we are getting the superior recombinant. Through the hybridization, what happens? What are the steps are involved in the hybridization? One is emasculation. In previous lesson we learned emasculation, bagging, another one is rebagging. For the conducting of these three processes, emasculation, bagging and rebagging, we have to wait until the flower or the plant get into the maturity stage. So whenever you wait for their maturity state, then what happens? It consumes more time. When the time is consuming, then what happens? We have to wait for many days or the many months, or it may be the years. So that is the reason why hybridization is a most time taking and tedious process. Okay. So this is about what? Cross hybridization among the selected one. So once again I am revising. Collection of germplasm, just desired characteristic seeds or the plants we are collecting. Next, evaluate and selection of the parental one. Evaluate means among the germplasm, among the seeds, we are selecting which one as a parental, male parent or female parent in the bisexual plants when we see. Next, third one, we have labeled A is the male and B is the female. A has some characters and B also some characters, desired characters, but both did not possess the same type of the characters. Okay. Same type of character means when you take the protein, highest content, there less, disease resistance low, there it is a high. Superior recombinant means what we got the hybrid through the cross between the A and B, that is called as a superior recombinant. It consisting of the A and B, both parents' characteristics. This hybridization is the most tedious and time taking process. Why? Because desired chromosomes may be present or may not be present, or out of the 100 or 1000 only one may perform cross fertilization. Next, hybridization is a most time consuming process. Why? Because emasculation, bagging and rebagging, these all are different techniques or the process to prevent self pollination, to promote the cross pollination, so that it is not so easy, cross hybridization. Next, I am going to explain about the selection and testing. It is one of the crucial step in the hybridization steps. Why? Because now we got a superior recombinant. We are assuming that is a superior recombinant. Whether the superior recombinants present or not, selection is very tough. Okay. So, that is the reason why I am explaining that fourth step is a very crucial step to evaluate whether the desired combination of the chromosomes or the genes are present or not. Okay. So, selection and testing is a tough job in the steps of the strategies or plant breeding. Next one is testing. What is next one? Testing and release. Testing and release. So, in this one you got a one superior recombinant, or I am instead of superior recombinant you can also write as a hybrid. You got a superior recombinant, or else what we are calling, we can also call as a hybrid, no? Yes. Now, this hybrid variety directly we cannot send to the farmers. Okay. There are many steps are involved to send the farmers. Why? Because after sending the farmers, if they not produce proper crop, then total process goes waste. That is the reason why it will be under the supervision of Indian Council of Agriculture Research Institute. So, first it will be sent into the Indian Council of Agriculture Research Institute. Indian Council of Agriculture and Research Institute, which under the supervision of this institute many steps will be performed. First one, it will be sent to the research field. It will be sent to the research field. In the research field, scientists, they test the seed. What seed? Hybrid variety of the seed will be tested by them. Then after, here ideal conditions are present, it can able to grow or not, they test. Ideal conditions means here fertilizers and irrigation, means whether it is observing, whether it is growing less amount of the fertilizer or more amount of the fertilizer. Whenever we supply the less amount of the fertilizer, can it be grow or not, they test. Another one is ideal irrigation techniques. Ideal irrigation technique means it has to grow or give us a crop under the less water conditions. Okay, that also will be tested by the ICAR. So, first research field it will be grown, then second step ideal conditions that seed possess or not, that will be seen. After ideal conditions, what do they do? Okay. India will be divided into 3 climatic zones, 2 to 3, more than 2. Okay. 3 climatic zones. Okay. Sorry. Soil climatic zone. What climatic zone? Soil. You have to write here. Soil. More than 2 or 3 soil climatic zone, that hybrid will be tested, whether it is growing or not. We have North India, South India, okay, like that. Then whether it is growing in both type of the climatic conditions, like that. Even soil, there are different zones. Not only soil climatic zone, there are 3 seasons: summer, winter and the rainy season. That crop or that seed growing in which season, that also will be tested. Okay. Then growing season. Fourth step is growing season. Okay, summer, winter, like that. Then after, fifth step. After that, after completion of these all steps, they sent to the local bodies, means local research center. Local research center means every district headquarter or mandal headquarter, what will be present? Almost that is NSC, National Seed Corporation. National Seed Corporation. This National Seed Corporation, again in the local areas, it perform the test. Then when it is in that test, when they got a proper productivity, now that will be circulated in the market, that will be sent into the market as well as to the farmer. Okay. So this many steps are involved to get the desired seed or the plant. Okay. So with this, whatever the steps are present with the in the strategies for the production of the desired characters are completed. So what are the steps? Collection of the germplasm, then evaluation, selection of the parental one, cross hybridization among the selected one, and selection and testing and release. So testing and release, ICAR, Indian Council of Agriculture Research Institute, will be involved more to get the desired seeds. Okay. In the local places, National Seed Corporation. Okay. These are the steps which are involved in the plant breeding. Now let us see the next concept. See here, after the independence, after the 1947, India has faced much problem to get the food production. Day by day, the population is increasing, year by year the population is increasing. To meet the needs of the growing population, the food facilities whatever we have, food grains whatever we had, those are not sufficient. So that is the reason why millions of the people were died. Even during the 1942-43 Bengal famine, what we have faced? Bengal famine we have faced. In that Bengal famine, almost 2.1 millions of the people were died. So that all over the India and the world, especially in India, the scientists they have conducted many experiments on the seeds and the plants in order to fulfill the needs of the growing population. Then finally, they brought a revolutionary change in the crop improvement, that is the green revolution. Okay. India is a agriculture based country. So here almost 33% of India GDP depends upon the agriculture only. Okay, what is that? 33% of the India GDP depends upon the agriculture only. Next, here 62% of the people are the farmers depending upon the agriculture. Okay, this number is very, very important. Question will be framed from this number regarding the GDP. When it will be, so green revolution after the 1947, after the independence, we got the green revolution. So India's GDP how much? 33% based on the agriculture. Okay. Next, 62% of the people are depending on the agriculture as they work. Okay. Next, here M.S. Swaminathan belongs to Tamil Nadu. M.S. Swaminathan, he is the father of Indian Green Revolution. Father of Indian Green Revolution. Okay. Next, Norman E. Borlaug is the father of the green revolution all over the world, worldwide. Very, very important, these two. M.S. Swaminathan is a father of Indian green revolution. Norman E. Borlaug is the father of world green revolution. See, we are learning the word, two words, one is green, another one is a revolution. Green means the word green related to the plants and its development. Okay. So then revolution, revolutionary changes means sudden changes. In a less time we have to get more product. So here less time, more yield. Less time, more yield. Those all insect resistant, pest resistant and microbe resistant. They must possess insect, pest and microbe resistant and fertilizer responsive. Fertilizer responsive. Next, more nutritional value. It must and should possess more nutritional value. So these are the characters which have developed by them during the time of the green revolution. So less time, more yield, insect, pest, microbial resistance, along with that stress resistant. Stress means water, salinity, temperature, those all comes under the stress resistance. It has to be withstand by the less or excessive water changes, and salt concentration increase and decrease also it has to be withstand. Okay, temperature, based on the temperature also it affects on the plant growth and development. So whatever we got through the green revolution, those all hybrid varieties possess these all characteristics. Okay, who is the father of the Indian green revolution? M.S. Swaminathan, belongs to Tamil Nadu. Norman E. Borlaug, father of green revolution, England, from the England country. Which country? From the England country. Next, these two number we have to remember. What are the improved varieties? If you see, just now only I said 1947, after the independence of the India, we have faced many problems regarding the food grain production. Then after 1960s to 2000, they have produced semi-dwarf varieties. Semi-dwarf variety. What are those? For example, now I will write here, 1 minute. Here, first one I am writing wheat. Okay. Next I am writing here rice. So the duration between 1960 to 2000, in these 4 decades of the time, the enormous improvement of the food grains production. So wheat, for example, if you take the wheat, that is 11 million tons to 75 million tons increased. Whereas rice, if you take 33 million tons to 89.5 million tons increased. Okay. When, which year? During the 4 decades of a time, that is 1960 to 2000. What varieties? These two semi-dwarf varieties. Semi-dwarf variety of the rice and wheat, whenever they used, the productivity of the grains has increased enormously. It has occurred through the green revolution only. So, then after there is no scarcity of the food grains after the green revolution. Bengal famine 1942. So, during that time what happens, 2.1 millions of the people were died in order to not getting of the food grains, there is no proper productivity, so that many people were died. So, for that they found a solution, that is a green revolution. Next, let us see about the wheat. See, now I am writing here wheat. So, what wheat? This is a semi-dwarf variety. What is it consisting of? What are the characteristics it possesses? It possesses here RH gene. RH gene means reducing height gene. Reducing height gene. So, due to presence of the RH gene, the wheat plant has grown 2 feet to 4 feet of the height only. Next, in less time, more yield. Then quality, nutrition also more. Quality, nutrition also more. Next, it is showing the all type of the resistance and fertilizer responsive. Fertilizer responsive. So, these all characters the semi-dwarf variety of the wheat we got. Now, what is the name of the gene? Actual gene is present in the wheat, that is Norrin10. Norrin10. It is the actual gene which is present in that Norrin10. Who discovered? Norman E. Borlaug, okay, from England. Okay. Which country? Mexico. Which country? In Mexico. So, here it is a semi-dwarf variety. Which variety? Semi-dwarf variety. What is the name of the gene? That is a Norrin10. And who discovered? Norman E. Borlaug. Where? From the Mexico. So, now we got semi-dwarf variety, there is a much production. Okay. Then after the semi-dwarf variety of the wheat, M.S. Swaminathan, through the gamma mutation, Lemma Roja 64 or Sonara 64 in India, he brought from the Mexico to the India, he cultivated in India. Then after Norman E. Borlaug discovery of the semi-dwarf variety of the wheat, our scientist M.S. Swaminathan, what he has taken here? M.S. Swaminathan, same wheat only. Lemra Roja 64, Sonara 64. Okay, this he brought from Mexico to India. Which mutation? Gamma mutation. Who brought? M.S. Swaminathan. In India, in Indian subcontinent, when you see Sonara sonalica, it will be grown in Indian subcontinent. Okay. So, he brought from the Mexico to the India by changing what changed? Through the gamma mutation he brought some modification. Based on that again this one only, what is that, Lemna Roja or Sonara 64, in the year of this is 1963, 1960 to 1963, in between in Indian our native one he has developed, that is a Sonara. In India it has grown. Then after this is about the wheat variety. Then if you take about the millets, what are the millets? Bajra, Jowar, okay, etc. These are also Indian varieties our scientists produced. So with this semi-dwarf varieties, due to the semi-dwarf varieties, the production of the rice grains are enormously increased. Then our fulfills, our growing population fulfills has increased. Their fulfillment, their discoveries, whose due to the Norman E. Borlaug and the in our country M.S. Swaminathan, our people has fulfilled their requirements. Then in my tomorrow's class, next will be continued.